Hey Team Bios, so yesterday in class, or I guess today in class, you will, because I'm recording this before the start of class, you will have already ordered the mitosis and cytokinesis cards into order. This screencast is describing every phase, giving names to the phases, and talking about what's happening to DNA, spindle fibers, the nucleus, and the chromosomes in all of the different steps. So you know from previous night's screencast that interphase takes up most of the cell cycle. So most of the time the cell is doing its living, that it's in interphase. An interphase is composed of 3 phases, that we called G1, which was growth or gap 1. Rewrite this. G1, which is followed by, growth, or what the heck? G1, which is followed by the S phase, which stands for synthesis phase, and what's happened in the synthesis phase, or the S phase, is the DNA is replicated. So there are 2 copies of every piece of DNA. And then G2, where again the cell, that's a 1, where the cell is growing and getting bigger and doing normal everyday life stuff. And then in mitosis and cytokinesis, I moved cytokinesis. It was over here in the note that you have, and I moved it. I apologize, Campbell. In mitosis and cytokinesis, well, we haven't talked about that yet. That's what today's screencast is about. Okay. So let's get down to it. So during interphase, G1, G2, and S phase, if we look at a cell, you can see that the DNA it looks rough. I mean, I know there's variation in the color here, but the DNA is kind of uniform, and it's because it's in its loose, uncompacted form. Is loose and uncompacted. This is called chromatin. Now, the DNA, when it's in this form, is coiled around proteins called histones, and it's in this loose, uncompacted form, so it's accessible by the machinery that's doing transcription and, well, just transcription, which will be followed by translation, which will lead to proteins being synthesized. So when it's like this, the DNA is accessible. When we start to, oh shoot, I guess I should have talked about before this all begins. Okay, so when a cell divides from 1 cell into 2 cells, we're going to end up with 2 daughter cells that are genetic clones of one another. What this means is that if you were to look at this cell and compare it to this cell, you would see that they're exactly identical. They have all the same DNA in each nucleus. So the whole goal of mitosis and then cytokinesis is to divide up the DNA and make sure that both daughter cells end up with all the DNA they need, as well as all the organelles and cytoplasm and cell membrane and such. Okay, so when we do that, when we divide up the DNA, this most important part, we need to access the DNA because it's locked up inside the nucleus, and we also need to wrap it up in such a way that it's going to be easy to split. Okay, so during the first phase of mitosis, which is called prophase, the DNA begins to condense. And you can start to see the chromosomes. individual is spelled with an AL, I think. So as you can see here in this picture that we have, the DNA, the nucleus goes from being kind of this pretty uniform density to start to look like it's full of spaghetti. And these are the chromosomes. Now, remember, in S phase, we duplicated all of the DNA. So as the chromosomes start to condense, they're actually in pairs where this arm of the chromosome is exactly identical to this arm of the chromosome. And we're going to talk about how that replication happens later on in the year. But it's important for you to know that all of this DNA over here is exactly the same as all of this DNA over here. And it's going to be different than the DNA that's here and here, or the DNA that's here and here. But the pair that, these are called sister chromatids. Again, don't really want to get into this right now. These two that I've drawn in pink are all the same, and the ones that I've drawn in blue are all the same, but they're not the same as the ones in black. Anyway, back to this. So the chromosomes start to condense, and then something that you can't see in the pictures but is drawn into the drawing images is that the spindle fibers begin to form. So the spindle fibers are composed of the cytoskeleton, specifically microtubules. And they're going to be responsible for pulling the chromosomes apart later on. And the last thing that happens in this phase is the nuclear membrane begins to dissolve. So you can see, again, in the drawing, not so well in the picture, but the nuclear membrane, this black thing right here, is beginning to dissolve, and the spindle fibers are starting to form. Now, the next phase of mitosis is called metaphase. And in metaphase, the chromosomes all start to align on this invisible line called the metaphase plate. I guess we can label that. It's kind of like the equator of the cell. It's usually in the center somewhere, unless you have an asymmetrical division. So you can see the chromosomes are lining up here. Our spaghettis are being aligned at the center, at the metaphase plate, this invisible line at the center of the cell, and they're pulled into position by the spindle fibers. So I guess we should write that down. Chromosomes align on the metaphase plate. That's AL. And they're pulled into place, as I said before, by the spindle fibers. And then the spindle fibers are also holding them in position here, and they're attached at the very center with a specialized attachment that we'll talk about later. And also, at this point, the nuclear membrane is totally dissolved. Okay, now, the most dramatic phase is called anaphase. During anaphase, when there's equal and opposite tension on all of the chromosomes, they are cut, they're split down the middle. And these sister chromatids, they're called, so this, remember, this chromosome, it was attached to this one before, and they were genetic copies of one another, so totally identical sequence of DNA here and here, as well as here and here, and here and here, and here and here. They are pulled apart from one another. by the spindle fibers in anaphase. So chromosomes are pulled apart by spindle fibers. Okay, and then we move on to the last phase, which is actually kind of two phases at the same time. This is telophase or telophase. You'll hear it pronounced both ways. Telophase is happening at the same time as cytokinesis. So remember, mitosis is concerned with the separation or the splitting of DNA. And cytokinesis is everything else. So the splitting, of the cytoplasm, as well as all the organelles. And included in the organelles would be the cell membrane. So this is a plant cell, you can tell, because it's a cell wall around it and it's kind of boxy. When plant cells divide, they actually form a cell plate in the center, which extends from the middle out. Animals undergo, it's called a cleavage event, where they kind of, like, imagine making a balloon animal. You can take a balloon that's like a long hot dog shape and then twirl it in the middle. That's kind of more what happens in animal cells. They're pinched and squeezed from the outside in. Okay, but we'll look at pictures of animal cells dividing as well as plants. So you can see here the cell plate forming a cell wall in the interior of this image. But kind of more importantly, the nuclear membrane is going to reform around the DNA. So nuclear membrane reforms. The DNA starts to loosen back up. So it begins to uncoil and form chromatin again. Kind of loose chromatin. And then the spindle fibers start to dissolve. And then at the same time that this is all happening, that this is happening inside the nucleus, the organelles have been, pulled and divided. So this cell has a little rough ER, this cell has a little rough ER, this cell has a little smooth ER, chloroplasts are on both sides, mitochondria is on both sides. So that's the cytokinesis part of all of this. So this is happening simultaneously with telophase, which is concerned with what's happening to the nuclear envelope and the DNA and the spindle fibers. Okay, ultimately, we are left with two genetically identical daughter cells, and they will once again enter interphase and potentially will divide again unless they enter G0 or G0, where they exit the cell cycle. So yeah, that is how cell division happens. Okay, that's all.